We have Professor Joseph Nicolazzo from Monash University. So I'd like to thank him for accepting this invitation. So he's in Australia, so it's late afternoon for him right now, and we are just starting the day. So thank you for being with us today. I'd like to give a brief introduction about Professor Nicolazzo. He's a professor and director of Center for Drug Candidate Optimization at Monash Institute of Pharmaceutical Sciences, and he's also the International Associate Dean of the Faculty of Pharmacy and Pharmaceutical Sciences. Joseph Nicolazzo graduated with a PhD in 2004 from Monash University in Australia, focusing on buccal mucosal drug delivery. He joined the Faculty of Pharmacy and Pharmaceutical Sciences at Monash University in 2007 as a lecturer, where he's now a professor and director of the CDCO, as well as the associate dean. In addition to leading the CDCO, which provides expertise in drug absorption, distribution, metabolism, and excretion to facilitate drug development, Joseph has a particular interest in understanding the role of drug transporters in the brain access of therapeutics in neurodegenerative diseases such as Alzheimer's disease and amyotrophic lateral sclerosis. He's the author of 115 peer-reviewed manuscripts and is regularly invited to international conferences in the field of BBB biology, including Gordon Research Conference, Barriers of the CNS, and International Conference on Cerebral Vascular Biology. Joseph is immediate past president of the Australian Pharmaceutical Sciences Association and an editorial board member of Journal of Pharmaceutical Sciences, International Journal of Pharmaceutics, Journal of Pharmacy and Pharmacology, and Fluids and Barriers of the CNS. He's a steering council member of the International Brain Barrier Society, an executive committee member of the Board of Pharmaceutical Sciences of the International Pharmaceutical Federation, FIP, and has been induced as a fellow of FIP. Using a suite of in vitro, in situ, and in vivo models, his research is broadly classified into understanding the factors affecting BBB transport of drugs and endogenous molecules, the involvement of intracellular trafficking proteins in facilitating molecular transport across the BBB, the impact of Alzheimer's disease and amyotrophic lateral sclerosis on BBB transport of drugs and endogenous molecules, and exploring molecular targets at the BBB that can be utilized to reverse cognitive dysfunction associated with Alzheimer's disease. So with this, I'm leaving the microphone to you, and I'm thanking you once more for accepting to be our guest today. And at the end of the seminar, we will have a question and answers and discussion session. Thank you.
Okay, thank you very much, Adam. Thanks for having me. Thanks for the invitation, and for going through the biography. Yeah, really good pleasure to be able to present some of the work that we've been doing to colleagues in Turkey. One day I would love to visit Turkey. I was just saying to Adam before that we have a very big Turkish community in Melbourne, and I used to work in an area of Melbourne which we used to call Little Turkey, as a pharmacist, and a lot of people used to talk to me in Turkish, so I learned a little bit of the Turkish language, but I'll do the seminar in English today, not to embarrass myself. So I'll talk today about the blood-brain barrier and its role in not so much in drug transport, but more in how it helps to move molecules, naturally occurring molecules, both into the brain and out of the brain, and how that's relevant for Alzheimer's disease and cognitive function, which is an area I've been working in. First, I thought I'd show you what it's like here. So for those of you who have been to Australia, you may have seen these sites, but we're a very lucky country, very big country, lots of beautiful sites. And if ever you do make the trip down to Melbourne, please let me know. We now do have Turkish Airlines flying into Melbourne through Kuala Lumpur, I think, or through Singapore. So it's a one-stop trip.
They're actually flying directly from Istanbul, Joe. Directly from Istanbul to Melbourne.
To Melbourne now. Oh, okay, right.
Yeah.
Well, there you go. Even easier. So, the talk is going to focus pretty much all on Alzheimer's disease, which many of you will know is the leading cause of dementia. And there's a number of hallmarks of the disease, mainly the accumulation of beta amyloid, which leads to the formation of plaques, which I show you here on the right-hand side. Also, inside the neurons, there's accumulation of tau or hyperphosphorylated tau, which leads to tangle formation within the neurons. And there's also more and more evidence that there's neuroinflammation occurring. So activation of microglia and astrocytes, which lead to an inflammatory cascade. And that can also lead to neuronal death. One of the areas that I've been interested in for some time is the notion that there's also reduced levels or reduced concentrations of docosahexaenoic acid or DHA, which is a naturally occurring fatty acid that we get from the diet. And it has a lot of beneficial effects in neuronal health and in brain health. So DHA, this is the structure of DHA. The things that it can do, it can reduce the activation of the microglia, which are the brain's immune cell, reduce the production of inflammatory cytokines, help with neuronal growth, axon growth, and reduce basically the production and release of beta amyloid, which is one of the major proteins accumulating in the brain. So DHA, it's not synthesized by the brain. The brain only synthesizes about 2% of the DHA that it requires. And so the vast majority of DHA that is present in the brain has to come from the blood, has to come from our diet. And so the thinking was at the time, perhaps the reason why DHA is lower in the brain is because it has lower ability to cross the blood-brain barrier. So the blood-brain barrier, many of you would know, is the vascular interface between the blood and the brain tissue, and it's formed by the endothelial lining of the microcapillaries that circulate or flow through the brain. And it's been estimated that there's 640 kilometers worth of these microvessels that are circulating through our brain. And so I looked at the map of Turkey because I like to sort of equate this. And from Ankara, it's the distance to Izmir, which is down south. So that's about the length of the capillaries that are circulating in the human brain. And the endothelial cells that line those microvessels, so if we take a snapshot of those capillaries, what we see is the traditional view of the blood-brain barrier, where we have the endothelial cells surrounding the microvessels, and then some accessory cells, which are really important in supporting that brain endothelial cell. And a number of transporters. I've just depicted here one of the transporters. And so getting back to the story about, is it possible that the trafficking of DHA is lower in Alzheimer's disease across the BBB? And is that part of the reason why brain levels of this naturally occurring fatty acid are lower in the disease? We thought we'd actually assess this, and we did this using what we refer to as the in situ transcardiac perfusion technique. So we, in this particular technique in mice, we isolate we basically open up the thorax of the mouse. We ligate the descending aorta and perfuse into the left ventricle the radiolabeled DHA, which travels up the aorta, the carotid artery, and then flows out of the jugular veins, which we cut. And so we look at both the brain and the perfusate concentration of the radioactive DHA and measure the rate of transport over a very short 1 minute period. And so what we observed in the APP-PS1 mouse model, which is a mouse model of familial Alzheimer's disease, and these are mice that are about 8 to 9 months of age that have cognitive deficit, is that the rate of transport of radioactive DHA, which is on the y-axis, was significantly lower in the APP, the Alzheimer's mice, relative to the wild-type mice. So there definitely was a blood-brain barrier dysfunction of some sort that was leading to reduced trafficking of this fatty acid into the brain. So why was this occurring? what are the potential mechanisms? So we had to look at what or how DHA gets trafficked from the blood into the brain, and DHA, this fatty acid I've shown in red, can use either passive diffusion to get across the blood-facing endothelial membrane, or some active transport proteins that are present on the apical or luminal face of the endothelial cell. But what we were more interested in is, how does this poorly water soluble fatty acid, once it gets through either by passive diffusion or active transport, how does it basically swim across this aqueous cytoplasm to get to the other side of the membrane? Because DHA is poorly water soluble, and it's not going to want to leave this lipid membrane and jump into a pool of water. So we thought there must be some sort of carrier protein that I've depicted here in blue that helps to shuttle the DHA from the blood-facing to the brain-facing membrane of the brain endothelial cell. So what could this potential carrier protein be? We thought, let's look at fatty acid binding proteins, which are a family of intracellular binding proteins that are expressed in all tissues that are involved in fatty acid movement or fat requirement. And we looked at these as possible targets for the DHA trafficking process, because DHA is a fatty acid. We need a binding protein. So in the name, fatty acid binding protein makes the most sense. But also because there was evidence in the literature that of the 9 isoforms, one of these fatty acid binding protein isoforms was decreased in the neurons of individuals with Alzheimer's disease. So we thought, if DHA utilizes this protein, could this protein, could the expression of this protein also be reduced at the blood-brain barrier? And could this be part of the reason why the fatty acid has lower brain uptake? So we went on a bit of a search to see, of the 9 FABP isoforms, which ones are actually expressed in brain endothelial cells. And so we used human brain endothelial cells, a cell line, an immortalized cell line, and we measured the gene of all of the 9 isoforms, and we only found that FABP 3, 4, and 5 were expressed at the gene level, and at the protein level, both in the human cells, but also in isolated mouse brain endothelial cells, fatty acid binding protein 5 was quite highly expressed. So it's expressed at 15 kilodaltons, which is the correct molecular weight for any FABP. So from this point forward, we focused solely on FABP5 as the possible protein that's trafficking the DHA across the endothelial cell. To see whether it was involved or not in, firstly, cellular uptake of DHA, we silenced FABP5 in the brain endothelial cells using siRNA, and looked at the cellular uptake of radiolabeled DHA over a 1 or 2 minute period, I think it was. And we saw a significant but only slight reduction in the uptake of DHA. So we thought, well, maybe it is FABP5 is involved, but maybe it's not the only protein involved in the uptake. But then we had access to knockout mice. So we were able to get some FABP5 deficient mice from Japan, and we isolated the brain endothelial cells from the wild type or FABP5 containing mice, or from the FABP5 deficient mice. And when we looked at the cellular uptake of DHA in either of those cell types, we saw that there was a much more dramatic reduction in the uptake of DHA when this particular binding protein was diminished. So that was great from an in vitro perspective, but we really wanted to know in vivo, does a lack of the binding protein, FABP5, significantly reduce the trafficking of DHA across the blood-brain barrier? And so to do that, we used that same approach that I described in my first slide, but this time we looked at the radioactive DHA trafficking, or BBB transport, in the FABP5 containing and FABP5 deficient mice over a 1 minute period. And you can see there that when we lack FABP5, there really is a 40–50% drop in DHA trafficking across the BBB. So this gave us some really good evidence that DHA does require this particular binding protein in order to be trafficked across the BBB. So to summarize this work, well, got a bit of a cartoon here. We have DHA on the blood-facing side. It binds to this binding protein, fatty acid binding protein 5, which traffics it across. When we lack this protein, such as in genetic knockout, we have less trafficking and therefore less exogenous DHA appearing in the brain side. However, the studies we had done to date were only looking at exogenous or the radioactive form of DHA. What we were really interested in is if we grew these mice up, these knockout mice up, do they actually have lower levels of naturally occurring DHA? And do they also have a cognitive deficit, given that DHA is meant to be neuroprotective? So that these were the studies that we then did after that. So with FABP5 mice that were about 3 months old, I think these were, we looked at the concentrations of naturally occurring DHA relative to an internal standard in the cortex of FABP5 containing and FABP5 deficient mice. And they did have a reduced level of, or reduced concentration of naturally occurring DHA, which was going along with the hypothesis. And so we thought we expect, therefore, that the mice that lack FABP5 should have a reduced cognitive ability. So we did a number of tests to assess this, but I'm only going to show you one or two. And the first one is the water maze. So some of you may be familiar with the water maze, and what it is, it's a method to basically help identify whether hippocampal brain cognition is intact or not. So what we do is we have a pool of water. So it's a circular pool. We fill it with water with a little bit of paint so that it's not clear for the mice. And we have a platform over here with a flag on it. And what mice want to do is try and get out of the water, find that platform, jump on the platform and get out of the water. And they use these external cues to find where they're swimming. So a mouse that has, we do some training, and a mouse that has intact cognition will be able to use the external cues and find the platform more easily. And so that's what we call the latency, the time taken for these mice to find the platform. And over a number of training sessions, what we can see is that the mice that contain FABP5, so the mice that have normal DHA levels, they're able with training to find the platform a lot more quickly than the mice that lack FABP5, the ones that have the lower DHA in their brain. And so it's just showing that by lacking this particular protein, having less DHA in the brain, there is a cognitive deficit. And the other test we used was the novel object recognition. And very simply, well, in this particular study, we train mice in a box. They have two objects that they get used to and they get bored of. And then the next time they're put in the box, we put a different object there. And mice are usually inquisitive and they'll spend more time sniffing or sort of walking around the new object if they have intact memory. If their memory is not so good, then they won't recognize that this box is a new object, and they'll just spend a similar amount of time between the old and the new. And from that, we can determine a discrimination index. So the discrimination index is really the time spent with the new object relative to the old object. For the FABP5 containing mice, those that have normal DHA levels, they're able to discriminate towards the new object a lot more than those mice that don't have FABP5. So another piece of evidence there that FABP5 is critical in the whole cognitive domain. So I kind of went off a bit of a tangent in terms of, I started off talking about DHA transport is reduced in Alzheimer's disease, and I've hopefully convinced you that FABP5 is critical for the DHA trafficking across the BBB. So our next question was, in Alzheimer's disease, is the reason why we're getting less DHA trafficked across the BBB because we have less FABP5 at the blood-brain barrier? So just as a reminder, this was the original data that I showed you before. In the APP PS1 mice versus the wild-type mice, we have about a 40% reduction in the trafficking of radioactive DHA across the BBB. We isolated the brain microvessels from these mice. So basically take out the brain, isolate the microvessels, and then use ELISA to measure the concentrations of the FABP5 protein in the microvessels. And what we saw is that in the Alzheimer's disease mice, the APP PS1 mice, there was about a 40% reduction as well in the levels or the concentrations of FABP5. So this doesn't mean that the reduction in FABP5 is leading to the reduced trafficking of DHA, but it's clearly it's an association, and it may be that the lower FABP5 is at the BBB is contributing to the lower DHA trafficking across the BBB. So if that's the case, if this is really the culprit for why we have less DHA in the brain, is there a way that we can rescue this? Can we upregulate FABP5 to bring it back to normal levels so that we can get more DHA trafficked across the BBB? So we, at this point, really didn't know what regulated FABP5, but we had seen in the literature that PPAR agonists were able to regulate fatty acid binding proteins in prostate. I think it was prostate cancer cells. So we thought, without any other information, let's try a number of PPAR agonists to see if we can increase the expression of FABP5. And we started off in vitro. And what I'll do is I'll just take you to the sort of the end message. After trying a number of different agonists, we tried PPAR gamma, PPAR delta, and PPAR alpha agonists. And it was the PPAR gamma agonists, particularly pioglitazone, which increased both the gene expression of FABP5 and to a lesser extent, the protein expression of FABP5 in vitro. So this is in the human brain endothelial cells. So that was a promising result. Does this lead to a functional change? Does this lead to more DHA getting taken up into these endothelial cells? So we looked over time at the uptake of radiolabeled DHA into these endothelial cells that were treated with vehicle or treated with pioglitazone. And at each time point, there was a significantly greater amount of DHA appearing in the pioglitazone treated cells relative to the vehicle treated cells. So all good from an in vitro perspective. What about in vivo? Does pioglitazone treatment increase BBB abundance of FABP5 and increase DHA trafficking across the BBB? And so what we did here is we treated C57 black 6 mice. These are healthy mice. They're not Alzheimer's mice. We treated them with pioglitazone for 1 week at 40 milligrams per kilogram. And after that week, we assessed the trafficking of DHA, radioactive DHA, using that in situ perfusion technique that I've been using for this presentation. And we saw a significant increase in in DHA trafficking across the BBB. And when we looked at the microvessels, the brain microvessels, we also identified about a almost a doubling of the FABP5 abundance in those microvessels. We found that this was specific to radiolabeled DHA. We looked at sucrose as a marker of BBB integrity. That wasn't affected by pioglitazone. So it suggested that the increase in DHA trafficking was not just due to disruption that was induced by the pioglitazone treatment. So in summary, what we've found here is that pioglitazone, I should say that FABP5 important for DHA trafficking. In Alzheimer's disease, FABP5 goes down. We could use pioglitazone to increase the amount of FABP5 and therefore DHA trafficking. We've shown that in healthy mice. What we haven't done is actually treat mice with the Alzheimer's disease genes to see whether we can restore FABP5 in a disease context and whether that improves DHA trafficking and improves cognitive function. But that's something we're interested in looking at. So that was the first part of the major part of the presentation. And in the last 10-15 minutes, I'm going to talk about a different story. So in this first presentation or first part, we're looking at trying to improve the trafficking of a naturally occurring molecule into the brain. The second part of the presentation is about trying to get something out of the brain and ways to improve efflux of a particular molecule. And that's all got to do with P-glycoprotein. So P-glycoprotein, or PGP, which many of you may know about, is a really important efflux transporter that's expressed at the blood-facing side of the brain endothelium. And it's really important, and us in the pharmaceutical sciences world know it as a real hindrance to drug delivery, because it often binds to drugs that we want to try and get into the brain. It binds to those drugs and effluxes them and prevents them from getting into the brain. So it's quite a challenge for CNS delivery. But the other role that PGP plays, which is less known within the pharmaceutical science world, is that PGP also effluxes amyloid beta from the brain. So amyloid beta, you may recall from the start of the presentation, is that peptide that accumulates within the brain. And there's evidence, and I'll show you this in the next couple of slides, that PGP is important for basically clearing it out of the brain. So it has a natural function as well, as you'd expect, given it's expressed at the BBB and was expressed at the BBB way before drugs came along. So here's a simplified schematic of what I was trying to say. So through some mechanism which is not fully understood, A beta is taken up into the endothelial cell from the brain-facing side and then binds to PGP at the blood-facing side and helps move the amyloid beta into the blood. And one of the studies to show that to show the role of PGP in this process was from 20 years ago, a group in the US. What they did is that they injected radioactive amyloid beta, both amyloid beta 40 and amyloid beta 42, the two different major isoforms of amyloid, into the brain. And then they looked at the efflux from the brain into the blood in mice that had PGP and in mice that were PGP deficient. And for both species, for both peptides, the 40 and the 42, there was a dramatic reduction in the clearance of amyloid when PGP was deficient. Now, this was a really exciting finding. Why it's exciting, particularly for Alzheimer's disease, is because there's evidence that the expression of PGP at the blood-brain barrier is lower in individuals with Alzheimer's disease relative to those without the disease. And it's also been shown so that's in patients, but also in mouse models where microvessels have been isolated from either mice that have a transgene for amyloid overproduction when the PGP is probed for in these in the microvessels from these mice, you can see it's much lower relative to wild type mice. And also this is some of our own work where we've used the triple transgenic mouse model. So this is a mouse model that has both the amyloid and the tau pathologies. When we've isolated the microvessels, we see a dramatic reduction in P-glycoprotein relative to wild type mice. So if sorry, what was I was going to? Yes. So if there is a reduction in PGP expression, we'd expect that the clearance of amyloid would be lower in, sorry, what I'm trying to say is we know that PGP is involved in the efflux of amyloid beta. We know that the levels of PGP are lower in Alzheimer's disease. The question is the efflux of amyloid beta decreased? The efflux from the brain into the blood, is that reduced in mouse models of Alzheimer's disease? And so there's been a number of studies that have demonstrated where if you inject radioactive amyloid beta into the brain and compare its clearance or efflux from the brain into the blood, that in so this slope refers to the basically the efflux or the clearance. This is what the clearance looks like in healthy mice. This is what it looks like in a sporadic model of Alzheimer's disease. So in this mouse model of Alzheimer's disease, a dramatic reduction in the BBB efflux or brain efflux of amyloid beta. This is a sporadic model of Alzheimer's disease. We've also looked at a familial model of Alzheimer's disease, the APP PS1 mice. And what we've seen is that the percentage of transport of iodinated A beta is reduced in the Alzheimer's disease mice relative to the wild type mice at both 2 minutes and 10 minutes. So there is, I guess, reduced efflux of amyloid beta from the brain, and there is reduced abundance of PGP at the blood-brain barrier. So it's likely that this reduction in PGP leads to this reduction in clearance. So if I go back a couple of slides, go back to this slide. If PGP is that critical in clearing amyloid beta and PGP is lower in Alzheimer's disease, can we upregulate PGP as a way of increasing the amyloid beta clearance from the brain into the blood? And could that be a therapeutic strategy for improving cognitive function in Alzheimer's disease? So going back. All right. So what we've been looking at is copper as a potential regulator of P-glycoprotein. And the reason for this is that copper has this 20, 30 years of research showing that copper is really critical for activating signaling pathways in neurons for general neuronal health. And those signaling pathways that copper regulates in neurons are also expressed at the at the BBB, and are also involved in some way to PGP regulation. So we thought, can we use copper modulating compounds to upregulate PGP as a way of increasing the amyloid beta efflux from the brain into the blood? So the molecule we used was copper ATSM. So it's not a new molecule. It's been out for a number of years, and it's actually been used in clinical trials for ALS or motor neuron disease. And the reason it's a good molecule is because normally if you try and deliver copper, it's charged. Its ability to get into cells is poor unless there is a copper transporter. But it's shielded by a lot of these carbons and nitrogens, which form a ring around the copper, and that makes it a much more permeable entity. And when it enters the cells through some redox chemistry, that then causes the release of the copper to then act on relevant signaling pathways. So we use this compound at various concentrations in our human endothelial cells and assessed whether the copper ATSM treatment increased the expression of PGP. So we did that by Western blot, and we saw a nice increase in PGP expression with our copper ATSM treatment. We then went to see whether that led to increased function of PGP. And to do that, we used rhodamine 123, which some of you may know as a really well characterized PGP substrate. And when we had copper ATSM treatment, we saw less accumulation of the rhodamine because there was more rhodamine being effluxed out of the endothelial cells. So both expression and function were increased as a result of copper ATSM treatment. So that's in vitro. Oh, and I should say, what's the mechanism? Actually, I won't talk about the mechanism. I'll go straight to the next part, which was more about we showed in the human brain endothelial cells where the copper ATSM had an effect, which it did. We were more interested to know, does it have an effect in primary brain endothelial cells from mice? Because ultimately we were going to be dosing the compound in mice, and we wanted to see whether there was this human to mouse relationship. So we isolated endothelial cells from mice. We then treated them with copper ATSM for 24 hours, and we saw increased protein expression of PGP, which was great, and also reduced accumulation of rhodamine 123, which, as I mentioned before, is a fluorescent probe for PGP. Reduction in accumulation means that there is more of the rhodamine 123 being effluxed out of the endothelial cells. So that was all in vitro. The next step was to treat healthy mice with copper ATSM, and we did that for 28 days. And we then isolated the microvessels from those mice and measured PGP expression by Western blot. Relative to the vehicle treated mice, the brain microvessels from the copper ATSM treated mice had a significant increase in PGP expression, which was in line with what we were expecting. And they also had increased function of PGP. And the way that we demonstrated this was through looking at the brain uptake of radiolabeled digoxin, which is another well-known substrate of P-glycoprotein. So we use the in situ perfusion technique that I described before. After treating mice with either vehicle or with copper ATSM, we perfuse the mice with radiolabeled digoxin and then remove the brain after a period of 1 minute. And in line with an increase in the expression of PGP, we saw that copper ATSM treated mice had a reduction in the brain uptake of digoxin. And that's because there's more PGP that is effluxing the digoxin, and as a result, less digoxin actually appears into the brain. And this was not, this was an effect that was specific to digoxin. We looked at sucrose, which is a marker of paracellular diffusion, as I mentioned before, and copper ATSM had no effect on that particular process. So in summary, we can use copper ATSM to increase the expression of PGP in healthy mice and increase the function of PGP. Can we use it in an Alzheimer's disease setting to restore that PGP dysfunction? And can that then lead to improved amyloid clearance and improved cognitive function? And this is what the final study was all about in terms of treating APP PS1 mice or wild type mice for 2 months with copper ATSM or vehicle, and then removing the microvessels, or actually firstly looking at cognitive function, which I'll talk about, removing the microvessels to look at PGP levels, and also looking at how much amyloid beta was present in the brain at the end of that particular study. So the first readout was looking at PGP expression. So there's a few things here. So this is the PGP expression in wild type mice that were treated with vehicle or with copper ATSM. And interestingly, in the wild-type mice, the copper ATSM didn't actually modify PGP expression like we saw with the C57 mice in the previous study. Importantly, in APP-PS1 mice, there was a reduction in PGP relative to wild-type mice, and this was significantly rescued by the treatment with copper ATSM. So we're really excited to see that the copper ATSM had the ability to restore or rescue the expression of, or the downregulation of PGP in the Alzheimer's disease mice. We looked at the amyloid beta levels after these treatments, and as expected, we don't see any human amyloid beta in these wild-type mice, because the wild-type mice don't express the transgene that produces human amyloid beta. That was expected. We see quite a high concentration of amyloid beta in the APP-PS1 mice that are treated with vehicle, but this is significantly attenuated or reduced by the copper ATSM treatment. So our treatment that was affecting PGP was also reducing amyloid beta accumulation in the brain. And the final study then was to see, well, does our treatment actually improve cognitive function? And so for this, we used the Barnes maze. And the Barnes maze is kind of similar to the water maze, but only there's no water in this case. This is sort of like a round table, and there are about 20 little holes around the edge. And only one of these holes has a tunnel that goes underneath the table. And the mice will want to try and find that tunnel because there's light on top of the table, like a bright light, and the mice don't like that. So they want to try and find the circle that has the tunnel so that they can escape from the light. And again, they use external cues to try and remember which one of those 20 or so circles leads to the tunnel. And so we treated our mice with all the different treatments and then put it through this particular test. And we measured the latency. And remember, the latency is the time taken to find the hole that leads to the tunnel. So the wild type mice in blue, you can see that they learnt where to find the correct circle over 4 days. That's in blue. In green is the APP-PS1 mice. You can see that there's quite a difference there. In terms of the difference in latency, it's going from about 80 seconds to 160 seconds. So they've really lost their memory. They do eventually learn, but you can see it's not at the same level as the wild type mice in blue. And then in red is our Alzheimer's disease mice that have been treated with our copper treatment. And you can see that has reduced the latency. They're now becoming quicker in finding that particular hole with the tunnel. And this is just another way to represent that. The latency you can hopefully appreciate relative to wild type mice, whether they're given the treatment or the copper. The APP-PS1 mice have a significantly higher latency. They take a lot longer to find that particular tunnel, and then that's reduced. It's not restored completely, but it is significantly reduced with the copper ATSM treatment, which was nice to see. So in summary, our copper ATSM treatment through a mechanism which I didn't talk to just to save some time, but it's through ERK phosphorylation, leads to increased mRNA expression of MDR1, which is the gene encoding for PGP, increased PGP abundance at the blood-brain barrier, and increased efflux of amyloid beta, which appears to have a disease-modifying effect. So with that, I'd like to acknowledge the members of the group who were instrumental in conducting the studies that I spoke about today, and some collaborators, as well as some our funding body, the National Health and Medical Research Council, which is our national funding body, and also ANZ Trustees, who's another philanthropic organisation. And if you're really interested in the blood-brain barrier or barriers of the CNS, we're co-hosting the Gordon Research Conference in June 2026 in New Hampshire, USA. So if you are interested, applications are now open, and yeah, look forward to seeing whoever's interested in New Hampshire. And with that, thank you, Adam.
Thank you very much. It was really very interesting and also very